So students, in this video I'll be discussing about the different genetic disorders. As I told you in the earlier video, the genetic disorders can be classified as Mendelian disorders. Now these are the Mendelian disorders I will be discussing. Now why they are called Mendelian disorders? Because the inheritance of these diseases follows the Mendelian pattern of inheritance, and it occurs due to mutation in a single gene. So first we'll be discussing about hemophilia. Hemophilia is also called a bleeder's disease because, or it is also called the royal disease because this disease is also prevalent in the royal family of England. Now you have studied last year in class 11 the number of factors. There are different factors which are responsible for clotting of blood. Now hemophilia occurs due to missing of factor number 8 and sometimes due to missing of factor number 9, the Christmas factor. So 4/5 of the hemophilic cases is due to absence of factor number 8 and 1/5 of the hemophilic cases is due to absence of factor number 9. Now these are basically proteins. Now absence of these leads to non-clotting of the blood. So whenever there will be a minor cut, there will be excessive bleeding, non-stop bleeding, and the person may even die due to that. So this particular disease, it is an X-linked recessive disease. It occurs due to a recessive gene. Now why it is X-linked? Because that particular gene is located in the X chromosome. Now in case of hemophilia, the cases may be of the following kind. Suppose, since it is an X-linked disease, suppose there is a normal chromosome and there is the chromosome which is carrying the recessive gene for hemophilia. Now in case a female is having excess chromosome, we consider it to be a normal female. If the female is X and XH carrying the recessive gene in one of the X chromosome, this female is called the carrier female. Why it is called carrier? Because she will not express the disease since it is a recessive, and recessive character, you know, it gets expressed in homozygous condition. In case of male, and if it is a hemophilic female, so this is a case of hemophilic female, it is extremely rare that such females they survive because they die in the fetal condition. It is extremely fatal stage. In case of males, presence of a single hemophilic gene, recessive gene in the X chromosome is enough to cause the disease because there is no corresponding allele in the Y chromosome. It is an X-linked character. So such males will be hemophilic. They will be hemophilic male, and if the males are having normal X chromosome with Y, then this is a normal male. So in case of male, there are no carriers. Now suppose we are asked that what will be the progeny if there is mating between a carrier female with a, suppose, a normal male. So carrier female, the condition will be XXH. Normal male, the condition will be X and Y. So if there is mating, the probable combinations will be like this. So it can be XX, XXH, XY and XHY. So this will be a normal female, this will be a carrier female, this will be a normal male, and this will be a hemophilic male. So these will be the progeny. They might ask you what will be the percentage of, if the question is what will be the percentage of hemophilic offsprings if a mating is carried out between carrier female and a normal male, the answer will be 25%, or 1/4 of the progeny have the chances of becoming hemophilic male. So this way questions we can carry out combination. So that is the case of hemophilia. How it happens? Due to absence of either this factor or this factor, the blood fails to clot and the person may die due to severe bleeding, even due to a minor cut. So that is the case of hemophilia, which is, we should remember, it is an X-linked recessive disease. The second disease that we are going to discuss is that of color blindness. Color blindness is also an X-linked recessive disease because it occurs due to a recessive gene present in the X chromosome. And what happens in color blindness? A person fails to distinguish between red and green color. This is due to absence of light absorbing pigments, formation of light absorbing pigments in the retina for these two particular color. So color blindness, inheritance of color blindness will be similar to that of hemophilia. And as we know, these are X-linked recessive disease, so it is very easy for such diseases to get expressed in the males, because in males, for the disease to be expressed, you just need one allele, one recessive allele in the X chromosome. But for the disease to get expressed in the females, you need both the genes to be recessive in both the chromosome, X chromosome. So such combinations are difficult to get, but such combinations are very easy. So most of the X-linked recessive disease are very prevalent among the males compared to the females. But however, the females can be carrier. So the inheritance pattern of color blindness is similar to that of hemophilia. The third one that we have, third disease, it is called the sickle cell anemia. Anemia, you know, it is less blood or less amount of hemoglobin present in the blood. So sickle cell anemia, normally we know that red blood cells are discoid in shape. They are biconcave discoid in shape, which helps them to move very easily. However, in sickle cell anemia, under some special conditions, the shape of the red blood cells becomes sickle shape. And when the cells become sickle shape, their flow is hampered, their oxygen carrying capacity is hampered, they may undergo clotting at certain position, and due to sickle cell anemia, the person may experience, under the lesser symptoms are experiencing fatigue and pain, and under severe condition it might lead to damage to the different organs as well. Now this sickle cell anemia, it is an autosomal recessive disease. Autosomal recessive disease, recessive, you know, autosomal means it is not linked to the sex chromosome, it is linked to other chromosome. Now what basically happens in sickle cell anemia, normally hemoglobin is formed by a normal gene, HbA. Now due to mutation, when this gene gets mutated, this is the gene for sickle cell anemia. Now what basically happens, it happens due to problem in the gene which codes for the beta globin chain. Hemoglobin has got alpha globin chain and beta globin chain. So what basically happens in the sequence which codes for this beta globin chain, there is a sequence or a triplet codon called GAG. Now due to point mutation, this A gets replaced with U. We are talking about RNA, that's why we are writing U. So GUG. So normally GAG, it codes for valine. It codes for glutamic acid. It codes for glutamic acid. Now when this A will get replaced with U, so this gets replaced with the glutamic acid, amino acid in the protein chain will get replaced with valine. So a single conversion or single change in the amino acid, suppose there is the beta globin chain, it is a protein, globin is a protein, it is made up of amino acid. Suppose this particular amino acid should have been glutamic acid, but instead of glutamic acid. This glutamic acid is replaced by which amino acid? It is replaced by valine. So you see a single amino acid changing in a particular protein chain, it changes the entire configuration of the cell. This happens in low oxygen concentration or so with height and all. So it can prove to be deadly. So how deadly mutation, even point mutation can be, from this example we can understand. There is another case or another Mendelian disorder which is called Phenylketonuria. Now, phenylketonuria is also an autosomal recessive disease. It occurs due to a change in chromosome number 12. Now, what is that change? Due to this mutation in this particular chromosome, there is this particular chromosome, actually that particular gene in this chromosome, it codes for a protein which is called phenylalanine hydroxylase. Phenylalanine hydroxylase. Now, from the name you can make out this is actually an enzyme. So this enzyme, the code for production of this enzyme is present in this chromosome, chromosome number 12. So when there will be a mutation in that particular gene which codes for this particular enzyme, this enzyme will not be produced. Now, what is the function of this enzyme? This enzyme actually helps in conversion of phenylalanine into tyrosine. This is the amino acid. So phenylalanine is converted into amino acid tyrosine with the help of phenylalanine hydroxylase. Now, when there will be a mutation in this particular gene in chromosome number 12 which codes for phenylalanine hydroxylase, this enzyme will not be formed. When this enzyme is not formed, there is accumulation of phenylalanine because it fails to get converted into tyrosine. And when this phenylalanine accumulates through some other pathway, it gets converted into phenylpyruvic acid, which gets excreted through the urine. Over long duration, it might lead to your failure of the kidneys as well. So presence of phenylpyruvic acid in the urine is a way to test the disease called phenylketonuria. It leads to in long run, it will lead to hamperment of development of the brain, and so these are the different types of Mendelian disorders: hemophilia, then we have colorblindness, sickle cell anemia, and phenylketonuria. One more is there, thalassemia. It will require a detailed description, so that I will be discussing in the next video. Thank you.